नमस्ते बिटिया, हमेशा खुश रहो बिटिया. I am Dr. Lily Trivedi from Jyoti Vidyapeeth Women's University, and today I am going to take a session on the lab of agriculture microbiology, and the topic session is methods of sterilization, nutritional media and their preparation. Now, what are the sterilization methods? The sterilization methods or disinfection, they can be either physical, chemical or physiochemical. In the physical methods, traditionally used sterilization methods are the sunlight, heat, vibration, radiation and filtration. The dry heat has always been used for sterilization. For example, we have the red heat, the flaming, the incineration, the hot air oven, and the infrared. The moist heat includes below 100 degrees centigrade, at 100 degrees, and above 100 degrees. And in the radiation, we have the ionizing and non-ionizing, and the electromagnetic particle. Filtration methods of sterilization include earthenware, asbestos, sintered glass, or membrane filtration. We also use chemicals for sterilization like alcohol, aldehydes, phenolics, halogens, heavy metals, and surface inactivatants, some dyes. In the gaseous forms, the chemicals are formaldehyde, ethylene oxide, and plasma. Now, when we come to the commonly used methods, in lab, dry heat sterilization is most common. It is in the form of an oven which is specially designed, and for 160 degrees centigrade for 1 hour is enough for sterilization. It is a suitable method for sterilization of substances by moisture. Now, the disadvantage is there is a long heating time and high temperature is required. We have another method, that is moist heat sterilization, and for which we use the autoclave. Now, autoclave uses the saturated steam under pressure at the temperature of 101 degrees centigrade for 15 minutes. Now, microorganisms are killed very efficiently and due to this high pressurized saturated steam. But for those materials which cannot withstand the temperature of 115 centigrade, for them this is a disadvantageous method. Sterilization by radiation is also involved. In these two techniques are involved. One, it leaves chemicals which form new compounds inside the microorganism. Second is the nuclear protein structure is destroyed inside the microorganism. For example, cobalt-60 is used for gamma ray sterilization process, and the isotope cobalt-60 is responsible for the disintegration of unstable atoms. Now, it kills the microorganisms by isolating atoms of essential substances which are present inside the cell. In the chemical methods, ethylene oxide is used. It is a special type of chemical sterilization where gases and vapor are used, and the gas is safe and non-inflammable. It is a widely used gaseous sterilization. As a disinfectant or decontamination, we use the preservation methods which prevent these microbes from spoiling the products like food products or pharmaceuticals. Sanitization means removal of microbes that are threat to food industry, water conditioning, and public health. So sanitizer is usually an agent which is presently being used very much, and that reduces the number of bacteria or microorganisms to a safe limit. Now, disinfectants like chemicals using alcohol, aldehydes, halogens, phenols, they are most commonly used for sterilization. Now, steam sterilization cannot be used everywhere. So, at those places, these chemical agents are very much useful. Only disadvantage is that there is 40 to 60% humidity is required for their use. In the mechanical method, for solutions to be sterilized only, we use the deep depth filters or screen filters. There, the filters can consist of particulate or microbial filters or final filters. They remove the microorganisms by the adsorption on the filter medium, and all the filter filling and sealing process in the filter screen filter should be done under septic conditions, and sterilization test is also required. Now we come to media. Nutrient agar media is a general purpose nutrient media for mostly for microorganisms, especially for bacteria. Now it is commonly used, and the basic constituents for growth of bacteria are present in this medium. We have 0.5% peptone, 0.3% beef extract or yeast extract, 1.5% agar, and 0.5% NaCl. The water used should be distilled water, and pH should be 7.4 and 25 degrees centigrade. The uses of nutrient agar: it is commonly used for isolation and purification of microbial culture colonies, used for producing the bacterial lawns or carpets for antibiotic susceptibility tests. It is also used for purity checking. It is used for teaching or demonstration purposes where we require a prolonged growth and ambient temperature. So all these are uses of nutrient agar, which is an important nutritional media for bacteria. Now the important commonly used media for fungus is potato dextrose agar. It is a general purpose. It can grow for yield, molds, etc. And it is recommended for plate count method. This is a nutritionally rich base for mold sporulation and pigment production. Now, potato infusion: 200 grams of potato and dextrose to 20 grams, agar 20 grams, and distilled water 1 liter. To prepare the potato infusion, we boil 200 grams sliced unpeeled potato in 1 liter distilled water. We filter it through cheesecloth and save the effluent for which is a potato infusion. Nowadays, commercially dehydrated form of PDA is also used. Now, we mix this dextrose, agar and water and boil to dissolve all the components. After that, sterilization is done in an autoclave at 15 minutes at 101 degrees centigrade. After this, 20 to 25 ml portions are poured into 15 into 100 Petri dishes, which are already sterilized before. The pH of the medium should be 5.6. So all my material has been taken from this. I thank you very much. I hope you found this lecture interesting, and if you have any query or any doubt, please write in the comment box and it will be resolved soon. This session was powered by the digital version 2.0 of Jyoti Vidyapeeth Women's University. Namaste. I hope you found this lecture interesting. Thank you.